শোনা যাচ্ছে? শোনা যাচ্ছে কি?
There's no sound.
Hi Shapanda, how are you? Shantanu here. You're on mute.
অনেকদিন বাদে দেখা হচ্ছে।
কেমন আছেন?
ভালো, ঠিক আছে।
অনিন্দকে তুলে নিয়ে এসছি। হ্যাঁ, ও কোথায় চায়না ফাইনালে কি করবে চায়নাতে পড়ে থেকে? হ্যাঁ, তো রাইট।
অনিন্দ আমার সঙ্গে এই ব্যাপারে কথা বলেছে।
আচ্ছা,
আই ফুললি সাপোর্টেড হিম।
অনেকদিন বাদে স্বপন দত্তকে দেখলাম।
হেমন্তদা কেমন আছেন?
এই আছি ভাই।
হেমন্তদা, আপনি আমাকে চিনবেন না বোধহয়, বহু বছর আগে আমি RKM এর ছাত্র, আপনার সঙ্গে অনেক কথা বলেছি।
হ্যাঁ, এই শান্তনু, দাঁড়াও, তোমায় জানো আমি কোথায় দেখি, তোমার বাড়িতে, তুমি কি ইয়েতে ছিলে নাকি?
হ্যাঁ, বাড়িতে এসেছিলেন আপনি, ব্যাঙ্গালোরে এসেছিলেন।
হ্যাঁ, ওই মালেশ্বরমে নাই?
হ্যাঁ, মালেশ্বরমে, দেখুন আপনার মনে আছে পুরো।
হ্যাঁ, ঠিক।
আপনি ছিলেন, ডক্টর পাকলাসি ছিলেন, আর RKM ছিলেন।
হ্যাঁ। তুমি একটু ওয়েট গেইন করেছো।
হ্যাঁ,
ঠিকই আছে, আমি তো একই দেখছি।
ওকে, আমি বহুদিন বাদে দেখলাম। আচ্ছা, তুমি তো RKM এর কাছে করেছিলে না?
হ্যাঁ,
মনস্যান্টোতে ছিলে না?
মনস্যান্টোতে ছিলাম বহু বছর। হ্যাঁ।
এখন কোথায় আছো?
এখন রিলায়েন্সে আছি।
ও, তুমিও রিলায়েন্সে?
হ্যাঁ।
তুমিও রিলায়েন্সে?
He's a big boss in Reliance.
আর অনিন্দ বন্দ্যোপাধ্যায়, এর সঙ্গে আমার আলাপ হয়নি ভাই।
অনিন্দর সাথে?
হ্যাঁ, অনিন্দর সাথে আমার আলাপ হয়। ও তো স্পিকার আজকে।
হ্যাঁ, অনিন্দ স্পিকার আজকে। অনিন্দ আমার টিমেই আছে ও। হ্যাঁ,
অনিন্দ আজকে মেইন স্পিকার।
হ্যাঁ,
অনেকদিন আগে আপনার সাথে আলাপ হয়েছিল।
আমার সাথে?
হ্যাঁ।
কোথায় ভাই?
কলকাতাতেই।
আচ্ছা। রীতা ঘোষকে দেখছি। রীতা কেমন আছেন?
কানেকশন চলে যাচ্ছে।
হ্যাঁ।
আমাদের হোস্ট কোথায়?
হোস্টটা একবার মুখখানা দেখেছিলাম।
আচ্ছা।
হেমন্তদা ভালো আছেন?
হ্যাঁ, আমি তো।
হ্যাঁ, ভালো আছেন?
হ্যাঁ,
সব ভালো
আছি ভাই। আচ্ছা, আমাকে কি দেখা যাচ্ছে তো নাকি? আমি এটা নিয়ে
আমরা আপনাকে দেখতে পাচ্ছি। আপনি হয়তো অতটা ভালো দেখতে পাচ্ছেন না নিজেকে, আপনার সাইডে থাকবে।
হ্যাঁ,
আমরা দেখতে পাচ্ছি।
আর নিজেকে দেখার দরকার নেই। অমিত ঘোষ কোথায় গেল? অমিত ঘোষের আসার কথা তো।
আসবে, ওর তো শেষে, লাস্টে বলবে।
হ্যাঁ,
কিন্তু 5টার সময় ঢুকবে।
সবাই কি সব যে যার নিজের বাড়িতে আছেন তো নাকি?
এই তো ভাইস চ্যান্সেলর এসে গেছে। হোস্ট ভাইস চ্যান্সেলর।
ভিসি স্যার কেমন আছেন?
তোমার মিউট করা আছে শঙ্কর।
সেই দাদা সবসময় গাইড করে, এটা হচ্ছে নিয়ম। সব ভালো, একটা জাস্ট সবাই ভালো আছেন তো? হেমন্তদা কেমন আছেন?
এই তো ভালোই আছি ভাই।
হ্যাঁ, স্বপনদা সব ঠিক আছেন?
সব ঠিকঠাক।
আচ্ছা, ভেরি গুড। আচ্ছা,
when active vice chancellor is taking charge, বাকিরা তো সব ঠিকই থাকবে।
সেই আমরাও আছি মোটামুটি সব, এই কোভিড সিচুয়েশনে যা অবস্থা, যাই হোক।
মেনে নিতে হবে ভাই, মেনে নিতে হবে।
হ্যাঁ, সেই তো আমাদের প্রোগ্রাম শুরু একটা করা যেতে পারে এখন। জাস্ট ওই প্রথমে একটা ভিডিও ব্যবস্থা হয়েছে, তারপর থেকে আমরা ঠিক 5টার সময় আমাদের টেকনিক্যাল সেকশনটা শুরু করব। তো সবাই আছে, আজ তার মধ্যে বেশ কিছু আসবে 5টার মধ্যে আর কি, সেই জন্য টেকনিক্যাল সেশনটা আমরা 5টার থেকে রেখেছি। এখন সেটা স্টার্ট করা যেতে পারে।
Sure.
স্যার ভিডিওটা স্টার্ট করে দেবো তাহলে?
হ্যাঁ, শিওর।
ভিডিও স্টার্ট করো।
The CRISPR-Cas9 system is a tool for cutting DNA at a specifically targeted location. The technique has already revolutionized gene editing, but scientists are always looking for new possibilities. So what else can CRISPR do? Since being discovered in a bacterial immune system, CRISPR-Cas9 has been adapted into a powerful tool for genomic research. There are two components to the system: a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes. But making double-strand breaks isn't all CRISPR can do. Some researchers are deactivating one or both of Cas9's cutting domains and fusing new enzymes onto the protein. Cas9 can then be used to transport those enzymes to a specific DNA sequence. In one example, Cas9 is fused to an enzyme, a deaminase, which mutates specific DNA bases, eventually replacing cytidine with thymidine. This kind of precise gene editing means you could turn a disease-causing mutation into a healthy version of the gene, or introduce a stop codon at a specific place. But it's not all about gene editing. Several labs have been working on ways to use CRISPR to promote gene transcription. They do this by deactivating Cas9 completely, so it can no longer cut DNA. Instead, transcriptional activators are added to the Cas9 by either fusing them directly or via a string of peptides. Alternatively, the activators can be recruited to the guide RNA instead. These activators recruit the cell's transcription machinery, bringing RNA polymerase and other factors to the target, and increasing transcription of that gene. The same principle applies to gene silencing. A CRAB domain fused to the Cas9 inactivates transcription by recruiting more factors that physically block the gene. A more outside-the-box idea for using CRISPR is to attach fluorescent proteins to the complex, so you can see where particular DNA sequences are found in the cell. This could be useful for things like visualizing the 3D architecture of the genome, or to paint an entire chromosome and follow its position in the nucleus. CRISPR has already changed the face of research, but these new ideas show that what's been achieved so far could just be the tip of the iceberg when it comes to CRISPR's potential. Whatever comes next, it seems the CRISPR revolution is far from over.
Hi, my name is Jennifer Doudna from UC Berkeley, and I'm here today to tell you about how we uncovered a new genome engineering technology. This story starts with a bacterial immune system. That means understanding how bacteria fight off a viral infection. It turns out that a lot of bacteria have in their chromosome, which is what you're looking at here, a sequence of repeats shown in these black diamonds that are interspaced with sequences that are derived from viruses. And these had been noticed by microbiologists who were sequencing bacterial genomes, but nobody knew what the function of these sequences might be until it was noticed that they tend to also occur with a series of genes that are often encode proteins that have homology to enzymes that do interesting things like DNA repair. So it was a hypothesis that this system, which came to be called CRISPR, which is an acronym for this type of repetitive locus, that these CRISPR systems could actually be an acquired immune system in bacteria that might allow sequences to be integrated from viruses and then somehow used later to protect the cell from an infection with that same virus. So this was an interesting hypothesis, and we got involved in studying this in the mid-2000s, right after the publication of 3 papers that pointed out the incorporation of viral sequences into these genomic loci. And so what emerged over the next several years was that, in fact, these CRISPR systems really are acquired immune systems in bacteria. So until this point, no one knew that bacteria could actually have a way to adapt to viruses that get into the cell, but this is a way that they do it. And it involves detecting foreign DNA that gets injected, like shown in this example, from a virus that gets into the cell. The CRISPR system allows integration of short pieces of those viral DNA molecules into the CRISPR locus. And then in the second step of that's shown here as Crispr Rna Biogenesis, these CRISPR sequences are actually transcribed in the cell into pieces of RNA that are subsequently used together with proteins encoded by the Cas genes, these CRISPR-associated genes, to form interfering or interference complexes that can use the information in the form of these RNA molecules to base pair with matching sequences in viral DNA. So a very nifty way that bacteria have come up with to take their invaders and turn the sequence information against them. So in my own laboratory, we have been very interested for a long time in understanding how RNA molecules are used to help cells to figure out how to regulate the expression of proteins from the genome. And so this seemed like also a very interesting example of this. And so we started studying the basic molecular mechanisms by which this pathway operates. And in 2011, I went to a scientific conference and I met a colleague of mine, Emmanuel Charpentier, who is shown in this picture on the far left. And Emmanuel's lab works on microbiology problems, and they're particularly interested in bacteria that are human pathogens. She was studying an organism called Streptococcus pyogenes, which is a bacterium that can cause very severe infections in humans. And what was curious in this bug was that it has a CRISPR system, and in that organism, there was a single gene encoding a protein known as Cas9 that had been shown genetically to be required for function of the CRISPR system in Streptococcus pyogenes. But nobody knew at the time what the function of that protein was. And so we got together and recruited people from our respective research labs to start testing the function of Cas9. And so the key people in the project are shown here in the photograph. In the center is Martin Jinek, who was a postdoctoral associate in my own lab. And next to him in the blue shirt is Krzysztof Chylinski, who was a student in Emmanuel's lab. And so these two guys, together with Inez Fanfara, who's on the far right, postdoc with Emmanuel, began doing experiments across the Atlantic and sharing their data. And what they figured out was that Cas9 is actually a fascinating protein that has the ability to interact with DNA and generate a double-stranded break in DNA at sequences that match the sequence in a guide RNA. And in this slide, what you're seeing is the guide RNA and the sequence of the guide in orange that base pairs with one strand of the double helical DNA. And very importantly, this RNA interacts with a second RNA molecule called tracer that forms a structure that recruits the Cas9 protein. So those two RNAs and the single protein in nature are what are required for this protein to recognize what would normally be viral DNAs in the cell, and the protein is able to cut these up literally by breaking up the double helical DNA. And so when we figured this out, we thought, wouldn't it be amazing if we could actually generate a simpler system than nature has done by linking together these two RNA molecules to generate a system that would be a single protein and a single guiding RNA. And so the idea was to basically take these two RNAs that you see on the far side of the slide and then basically link them together to create what we call a single guide RNA. And so Martin Jinek in the lab made that construct, and we did an experiment, a very simple experiment, to test whether we truly had a programmable DNA cleaving enzyme. And the idea was to generate short single guide RNAs that recognize different sites in a DNA molecule, this circular DNA molecule that you see here. And the guide RNAs were designed to recognize the sequences shown by the red bars in the slide. And the experiment was then to take that plasmid, that circular DNA molecule, and incubate it with two different restriction or cutting enzymes, one called SAL1, which cuts the DNA sort of upstream at the far end of the DNA in this picture in the gray box, and the second site being directed by the RNA-guided Cas9 at these different sites shown in red. And very simple experiment. We did this incubation reaction with plasmid DNA, and this is the result. And so this is what you're looking at is an agarose gel that allows us to separate the cleaved molecules of DNA. And what you can see is that in each of these reaction lanes, we get a different sized DNA molecule released from this doubly digested plasmid that in which the size of the DNA corresponds to cleavage at the different sites directed by these guide RNA sequences indicated in red. So this was a really exciting moment, actually. Very simple experiment that was kind of an aha moment when we said we really have a programmable DNA cutting enzyme, and we can program it with a short piece of RNA to cleave essentially any double-stranded DNA sequence. So the reason we were so excited about an enzyme that could be programmed to generate double-stranded DNA breaks at any sequence is because there was a long-standing set of experiments in the scientific community that showed that cells have ways of repairing double-stranded DNA breaks that lead to changes in the genomic information in the DNA. And these, so this is a slide that just shows that after a double-stranded break is generated by any kind of enzyme that might do this, including the Cas9 system, those double-stranded breaks in a cell are detected and repaired by two types of pathways. One on the left-hand side that involves non-homologous end joining, in which the ends of the DNA are chemically ligated back together, usually with introduction of a small insertion or deletion at the site of the break. And then on the right-hand side is another way that repair occurs through homology-directed repair, in which a donor DNA molecule that has sequences that match those flanking the site of the double-stranded break can be integrated into the genome at the site of the break to introduce new genetic information into the genome. And so this had given many scientists the idea that if there were a tool or a technology that allowed scientists or researchers to introduce double-stranded breaks at targeted sites in the DNA of a cell, then together with all of the genome sequencing data that are now available, where we know the whole genetic sequence in a cell, and if you knew where a mutation occurred that causes a disease, for example, you could actually use a technology like this to introduce DNA that would fix a mutation or generate a mutation that you might like to study in a research setting. So the power of this technology is really the idea that we can now generate these types of double-stranded breaks at sites that we choose as scientists by programming Cas9, and then allow the cell to make repairs that introduce genomic changes at the sites of these breaks. But the challenge was how to generate the breaks in the first place. And so a number of different strategies had been produced for doing this in different labs. Most of them, and I'm going to show two specific examples here, one called zinc finger nucleases and the other tau effector domains. These are both programmable ways to generate double-stranded breaks in DNA that rely on protein-based recognition of DNA sequences. So these are proteins that are modular and can be generated in different combinations of modules to recognize different DNA sequences, requiring, so it works as a technology, but it requires a lot of protein engineering to do so. And what's really exciting about this CRISPR-Cas9 enzyme is that it's an RNA programmed protein. So a single protein can be used for any site of DNA where we would like to generate a break by simply changing the sequence of the guide RNA associated with Cas9. So instead of relying on protein-based recognition of DNA, we're relying on RNA-based recognition of DNA, as shown at the bottom. So what this means is that it's just a system that is simple enough to use that anybody with basic molecular biology training can take advantage of this system to do genome engineering. And so this is a tool then that really, I think, fills out an essential, previously missing component of what we could call biology's IT toolbox that includes not only the ability to sequence DNA and look at its structure. We know about the double helix since the 1950s. And then in the last few decades, it's been possible to use enzymes like restriction enzymes and the polymerase chain reaction to isolate and amplify particular segments of DNA. And now with Cas9, we have a technology that enables facile genome engineering that is available to labs around the world for experiments that they might want to do. And so this is a summary of the technology. It's a two-component system. It relies on RNA-DNA base pairing for recognition. And very importantly, because of the way that this system works, it's actually quite straightforward to do something called multiplexing, which means we can program Cas9 with multiple different guide RNAs in the same cell to generate multiple breaks and do things like cut out large segments of a chromosome and simply delete them in the same in one experiment. And so this has led to a real explosion in the field of biology and genetics, with many labs around the world adopting this technology for all sorts of very interesting and creative kinds of applications. And this is a a slide that's actually almost out of date now, but just to give you a sense of the way that the field has really taken off. So we published our original work on Cas9 in 2012. And up until that point, there was very little research going on CRISPR biology anywhere. It's a very small field. And then you can see that starting in 2013 and extending until now, there's just been this incredible explosion in publications from labs that are using this as a genome engineering technology. So it's been really very exciting for me as a basic scientist to see what started as a fundamental research project turn into a technology that turns out to be very enabling for all sorts of exciting experiments. And I just wanted to close by sharing with you a few things that are going on using this technology. So of course, on the left-hand side, lots of basic biology that can be done now with the engineering of model organisms and different kinds of cell lines that are cultured in the laboratory to study the behavior of cells, but also in biotechnology, being able to do to make targeted changes in plants and various kinds of fungi that could be very useful for different sorts of industrial applications. And then, of course, in biomedicine, with lots of interest in the potential to use this technology as a tool for, really coming up with novel therapies for human disease, I think is something that's very exciting and is really something that's on the horizon already. And then this slide just really indicates where I think we're going to see this going in the future with a lot of interesting and creative kinds of directions that are coming along in different labs, both in academic research laboratories, but also increasingly in commercial labs that are going to enable the use of this technology for all sorts of applications that many of which we couldn't have even imagined even 2 years ago. So very exciting. And I want to just acknowledge a great team of people that have been involved in working on the project with me, and we've had the terrific financial support from various groups as well. And it's been a pleasure to share this with you. Thank you.
The ability to cut the DNA where you want has revolutionized the life sciences. We can now easily edit genomes as desired, something that before was hard or even impossible. Today, CRISPR-Cas9 is a common tool in biochemistry and molecular biology labs. It's also used in plant breeding and for novel treatments of human diseases.
I feel extremely honored to receive this highest recognition from Sweden. I think surely of my former team in Vienna and at Umeå University who contributed to this great discovery. I also think of all my colleagues of the CRISPR field and all the scientists who have really done a fantastic job to bring this technology further that allows now to have a genome engineering technology, a very versatile one that is used by multiple scientists all over the world to modify genomes of cells and organisms. So it's a great discovery, and I'm very happy to be one of the pioneers. I find it for sure extremely nice that the award is going for 2 women. I think this is the first case ever seen for the Nobel Prize. I think it's quite also reflective of what you see nowadays. One has always to keep in mind that in general, Nobel Prizes are awarded 20, 30 years after the discovery. And this discovery is only 8 years ago, and it has bloomed extremely. So everyone is using now the CRISPR-Cas9 technology, and I think it's just reflective of what is happening in our days, meaning that scientific work involves collaborations, and we collaborate with scientists we think we should collaborate with, whether they are female scientists or male scientists. Here it's just happened that it's 2 female scientists, and it's quite reflective of what is happening in our days. I hope it will remain as such, but it's reflective of the fact that science becomes more modern and involves more female leaders.
One of the great things about saliva is it's pretty easy to collect. We don't need to have personnel in full personal protective equipment doing the collection. A key question right now is, will saliva be as good a matrix for testing as nasal swabs have been? And that's a question that we hope to answer with this study that we're running. So we're currently doing work to identify, first of all, how much viral shedding happens in saliva compared to in the nose, and then to figure out whether the actual saliva test that we're running is as sensitive as what we can detect when we're using nasal swab.
Thank you very much, Akash, for the presentation. I am the coordinator of ENVIS in University of Kalyani, and I welcome you all to this evening's webinar program, which is expected to be an exciting one. We shall now have the address by our Honorable Vice Chancellor, Professor Shankar Kumar Ghosh. He is an eminent biotechnologist himself, and is especially known for DNA barcoding of organisms. He has been behind many new developments in our university, especially in the fields of technology and with special reference to biotechnology. I now request our Honorable Vice Chancellor to say a few words before we begin the main program. Our Vice Chancellor, sir.
Good evening, all of you. Thank you, Professor Prasun Chakraborty. Prasun Chattopadhyay, rather, is the coordinator of our environmental biotechnology resource partner, Government of India. We have a very good ENVIS Biotechnology Center. I also, first of all, we welcome all of the participants as well as the resource person in this webinar, because all of we know that we need to develop ourselves is more in the promotion of science. Today is the International Students' Day coined by APJ Abdul Kalam, and today also we have start this promotion of science. In collaboration of NACI and INSA, I really thank to the executive members and conveners of these two academics, and also I thanks to the main speaker today, Anindo, who has spent a lot of work in this area, and our mentor cum teachers, Professor Amit Ghosh, he is always guiding us from the beginning, and Professor Hemanto Majumdar, and Professor Shapon Dotto, who is a long-term association, particularly from my side. I thanks all of you to join in this program. And also I thanks to Rita Ghosh, who is also DST coordinator in our university. It is one of the excellency of this program in India. Rather, I can say out of 5 excellency DST parts that we got under her leadership. I also thanks to my dean. 2 deans are there, and Dean Science, Indrajit Lahiri, and Dean Arts, Subhash Chandra Sarkar. Also, I have seen the Director of Distance Education, Professor Manush Babu Amader Manush Adhikari, and I have found my friend come senior at Bose Institute, Shantanu Dasgupta. Always I want to talk to him. It's very nice and discuss this issue also. So all of you, I thank once again. And today, why you want to thinking this kind of promotion of science, like 1.5 hour or not more than 2 hours. And this time, the COVID situation, we are very close to only the virtual mode, not in the face-to-face interactions. Really, it's a very unfortunate. However, we have to do. And our university is trying to get this kind of webinar in every time. And we have 48 affiliated colleges. And regularly, we are doing this kind of webinar so that we can interact our student, as well as we can give some kind of anti-depression to our student. So our university, the Kalyani University, started in 1960. So we have right now the 37 departments, and the last time we have created 5 new postgraduate in our university, like Genome Science, which is a second time in India, and MS in Data Science, which is the first time in India, and Nanoscience, Nanotechnology, Food and Nutrition, Computer Science. And there are another 5 departments we are going to develop near future. Already the highest body has already approved these courses, like actuarial science, molecular medicine, performing arts, journalism, etc., that we are going to start very shortly. So our university has university have started a new orientation program or new program in our part. Like we have started some of the new program, like academic festival. The university has a interdisciplinary approaches. We develop one academic festival, like 3 or 4 days. All the students, all the stakeholders came together and exchanged their views. And this is the first kind of academic festival, which it is a one kind of new part. And also in our university have started the career hubs, entrepreneurship of career hubs. We have achieved like a 15 crores of money to develop the entrepreneurship or like incubator. So we want to train them. There are different kind of 15 centers we want to develop over here. And we also made a research innovation that today, today in a noon, we had a great discussion about the research and innovation building. We have brought 15 crores of money to build one building, which all the research, innovation, and interdisciplinary program will be focused in a single building, that more than 40,000 square feet, with a very with a green technology that we have to build this part. And we have a 35 crores of money that we want to get some kind of mission mode research on the genomics, on the environmental issues, on the language lab, and so many things that we want to start with one umbrella in the research innovation program. And also we have, we will be happy that our university has recognized as a open university this time. So Government of India has started, UGC have started the open university in few areas that we have also going to start some kind of new things in my university. Like in Bengali department in our university, that they have their 50-year golden year this time. So we have to start right now online Bengali. Masters in online Bengali, which might be the first time in the world, I can tell you. And today we have submitted all the program. So expecting that we can initiate the online courses on Bengali. And another, we had to start the online courses of computer science. So the transition has come up in that way, that we want to initiate some kind of online program, online courses in our university. And also we try to build up the collaboration of international collaboration, and we have a very good ecosystem in Kollani. All of you know that there are very good institutes like NIBMG, AIMS, then ISAR Kolkata, Triple IT, Engineering College. So we try to make one Kollani economic hub or Kollani educational hub in near future. We have a planning, we are discussing this issue with other directors or vice chancellors so that we can build a one educational hub at Kollani. And all of you know that these are the very good place to educate our student, educate our research scholar. We are trying to make collaboration to each other. And I am very sure that University of Kollani will give a very good flagship in near future. And for that reason, our faculty members, officers, all are trying to get the flagships of this university. And this promotion of science is one of part that we are doing every time on this issue. So once more, again, I thank all the participant and expert today. And there are so many viewers on the YouTube, and it will continue. So in these few words, I again thanks all. With these few words, I back to Professor Prasun Chattopadhyay. Thank you.
Thank you very much, sir. We are always at the receiving end of the benefits of your enterprising and enthusiastic leadership. We now request our very able Dean of Faculty of Science, Professor Indrajit Lahiri, to say a few words.
So, Honorable Vice Chancellor, learned speakers, and other dignitaries, and the participants. The DST PARS 2 program, in collaboration with other agencies, is regularly organizing webinars and workshops on different scientific topics. This time, DST PARS 2 program joins its hands with the ENVIS Biotechnology Center of our university, NASI, and INSA. Usually, DST PARS 2 program organizes webinars and workshops on the topics of current interest. This year, the technology of so-called genetic scissors has won the Nobel Prize. So far, we the common people were not aware of this technology. But after the announcement of the Nobel Prize, a great attention of the media is focused on this technology. Naturally, the common people have become curious about the novelty of genetic scissors. Thus, it is a great endeavor of the DST Pars-2 program to organize such a webinar for popularizing scientific topics of current interest. In fact, through these endeavors, the DST Pars-2 program is doing excellent work with which it is able to place itself among the top 5 DST Pars programs in India. I sincerely wish a great success of the webinar and hope that the listeners and the participants will enjoy the lectures of the learned speakers who are the experts in the field of today's topic. Thank you. Thank you all.
Thank you very much, Professor Lahiri, for your very kind words. May we have Professor Rita Ghosh, who is the coordinator of the DST Pars-2 programs and also a convener of today's webinar, to say a few words. Professor Ghosh, please.
Thank you, Professor Chattopadhyay. Very good evening to all of you. Professor Shankar Kumar Ghosh, Honorable Vice Chancellor, University of Kolani, Professor Introductory Lecture, Dean, Faculty of Science, Professor Subhash Sarkar, Dean, Faculty of Arts, Professor Ashutosh Chattopadhyay, the Director of Invest Center, University of Kolani, and all the distinguished speakers of today, Professor Hemant Majumdar from IICB and Senior Nasi and NSA Scientist, Professor Shapan Kumar Dotto, former Director of ICAR, Professor Ananda Bandyopadhyay of Vice President of Reliance Industries, Professor Amit Ghosh, former Director of INTEC and NASI Chair Professor, all other distinguished participants of this seminar, and all students and scholars and colleagues who are here today in this webinar. I extend a very warm welcome to you on behalf of the DST course as well as on my personal behalf. The idea of having a series of lecture and promotion of science was first mooted by our Honorable Vice Chancellor. He said that during this period of COVID-19, when the university is closed down, let's have a series of lecture on promotion of science from DST first, so that you can enthuse the students and keep them active. And what a better start we could have when we could begin this series of lectures in collaboration with the Invest Center of Kolani University and Nasi and INSA, by beginning with a talk on genome editing by CRISPR-Cas9 system. So this has grabbed attention of everyone across the globe because this year's Nobel Prize in Chemistry has gone to this very important technique, which is, I believe, it's going to revolutionize therapeutic techniques in recent times, and it has wide application even as a diagnostic is used as a diagnostic technique. And very recently, as we saw that Professor Jennifer Doudna, she has developed a very simplified technique based on this, which could detect COVID-19 in 5 minutes. So it's just a modification of this one to give us a very fast and cost-effective technique. Our very own CSIR IB is also developed the Feluda, which is also based on this CRISPR-Cas9 technique. So I think today we are lucky enough to have with us Professor Anand Bandyopadhyay, who I've heard that has worked with Professor Jennifer Doudna. So it's really going to be exciting to hear from him. We are indeed lucky to have it with us today. DST Course has been, always endeavoring to kind of bring, to organize seminars, lectures, or workshops in different frontline areas. And just last month, we had a workshop on prevention, detection, and therapeutics of COVID-19, which was a very grand success because from the number of feedbacks that we got, it was really very exciting to know that it has been beneficial. People have enjoyed this. Even DST Pers, they also members of the DST Pers were present during during the first year of the seminar, the workshop, and they too mentioned it in the review meeting, which was held a few days later. So I think today's lectures is going to be also equally successful. I would not keep you waiting. I am happy listening to all of you. And in advance, I would like to thank all the speakers for being here today. Thank you.
Thank you very much, Professor Ghosh, for your nice speech. It's now my turn to say a few words. I represent the Envis Center of the university. This center was started in our university by Professor Subhash Chandra Shatra in the mid
Sir, you are not audible.
We cannot hear you, Professor Chattopadhyay.
Oh, maybe he will rejoin. Okay, might be he will rejoin. So from let me start. শোনা যাচ্ছে?
স্যার শোনা যাচ্ছে স্যার।
Let me start. He may join later on. So, as the University of Kolkata, this time of promotion of science will be going and in association of NASI and INSA. So, from our university size, we are giving some kind of information. Now, I the program with collaboration of NASI, and he is the Professor Majumdar is a renowned scientist and served through his life on the basically molecular biology, and he worked in several areas in particularly Lithuania, Donovany, and given more than full life basically on the science, and he was very much associated from when I was joined in IICB in 1995. From that point of time, I was associated to him, and he always guide us. Honestly, I am telling, he always guide us. He's a local chapters of the NACI, convener of the NACI and INSA. So I request Professor Mojumdar to speak few words in this connection of this program. Professor Mojumdar.
Can you hear me?
Yes, 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 sir.
First of all, I thank the university, particularly Professor Shankar Ghosh, the Honorable Vice Chancellor of this university. I'm very happy that he has invited me and also requested me to be associated with this program on promotion of science in the area of genetic seizures. Today, there will be a lot of discussions. Many speakers have come. I am representing INSA and NACI also, because I am the president of NACI Calcutta local chapter and also the convener of INSA, Indian National Science Academy. These two academies are the most important academies because, but before that, I would say that National Academy of Science is the oldest academy in the country. It was founded by Sir Meghnad Saha at Allahabad. So he was a professor of physics at Allahabad University, and he felt during that time, he felt that post-Indian, post-World War, there is a real dearth of knowledge, and that knowledge has to be propagated to the entire nation. So we formed the committee, and later the committee became very active, and NACI was formed. Similarly, INSA, most important academy, the most recognizable academy in the country, was established in 1935. Both these academies have the same charter, more or less the same. The charter is to educate people, to recognize people, and to spread science and technology to the underprivileged, mostly the underprivileged. So there are several activities are there, and in Calcutta local chapter, we are really actively working on that. We are holding several programs, but because of this COVID, we did not stop the program, scientific awareness program. We are still continuing, but alternatively, with the help of the webinar, we are organizing several programs. I'm very happy today. I have seen the picture of a building in this today. And the building is University of California at Berkeley. The name of that building is Stanley Hall. And I am very fortunate that I was there for 2 years working at Stanley Hall, and my next door scientist was another lady, and later she got the Nobel Prize. Her name is Elizabeth Blackburn. She was very famous for discovering a telomere, the end of the chromosomes. So on one hand, Elizabeth Blackburn got the Nobel Prize, and on the other hand, recently, Jennifer Doudna, also working from the same Stanley Hall where I used to work, they got the Nobel Prize. So thank you. And also I have seen another name, University of Zurich. I also spent 3 years in the University of Zurich. I learned that government DNA technology. So anyway, I'm fortunate and I'm really honored to become the president of the National Academy of Sciences in Kolkata chapter. My friends, respectable, my senior, Professor Amit Ghosh is there. Dr. Shopon Dotto is a very, very good friend of mine. And I can see the galaxy of scientists here who will present their work. And I am not going to take much time. I'm always with Vice Chancellor, Sir Shankar Ghosh. One point of time, I was his sir. Now he is the sir of everybody. So thank you very much. And I will enjoy the meeting. But I would request to Professor Shankar Ghosh that I have to leave a little early. I have another webinar in the German consulate. I have to deliver a lecture on in this infectious disease. So thank you. Please go ahead with your seminar. Anindo Bandyopadhyay, and then Chopon Dotto and Amit Ghosh. These three speakers, I will not be able to listen to those lectures, but anyway, I know what they are working. But I will listen to Anindo Bandyopadhyay sometime somewhere. But in my laboratory, CRISPR-Cas technology is being used for regulating the genomes of some of the genes of Leishmania. Thank you very much.
Sir, unmute করতে হবে। Thank you, sir. You have given a very enlightened of the Nassian INSA and your work. I really appreciate your presence in the today's program, because I know that you are very busy today. After that, you have a two webinar. One is German consulate and one is another international Leishmania group. However, you have joined and given your few words that really helpful for inspiring of our viewers. Professor Ashok Babu is there. You are joined. Professor Ashok. So you just you can give us few words about today's program.
Thank you very much, sir. I don't know how I got out of this this webinar somehow. Okay, I was talking about how the INVIS Center was set up in the university, and then after Professor Shatra retired some time ago, it kind of languished a bit. Then after the current Vice Chancellor took over, a new and very energetic coordinator was put in the place. He was professor of zoology, Professor Ashish Panigrahi. So under his leadership, a lot of activities of the INVIS Center came up, and it got national recognition also. Unfortunately, Professor Panigrahi moved because of career options to another university as Vice Chancellor. So the Vice Chancellor very kindly requested me to act as coordinator. So that's how I'm in the picture now. Very recently, I've joined as coordinator. So this INVIS Center is one of the 25 INVIS hubs of this country, and is now very well placed. We have 4 staff. We have lots of activities. There are webinars. There are special days being observed. There are booklets and other issues that are published. And as the current Vice Chancellor is quite visionary, he has many ideas, and we shall definitely work together to enhance the prestige of the INVIS Center in our university and make it really effective. That's all I would like to say, because we have many interesting speakers and a lot of very eager participants waiting for the main program. Thank you very much, sir.
Now we are in time. Now we have to start our technical program. So the inaugural part of this today's webinar, we are now going to the technical part. There are one, just want to share the first program, the introductory lecture, then main speaker, and then the lecture. Then there is a question answer session. So we have set in that way. So introductory lecture or will be given by our beloved plant biotechnologist, Professor Shapon Kumar Dotto. He has a quite wide experience in biological science, including crop science and particularly today's program, you know, I met and I few committees where I am, and he are the members. You know, when we discussed several issues, and he always to try to figure out that new technology or genome editing technology is the future of not for food security, but also in the other issues like health issues and other part on that. And he's a fellows, and he worked in a ICR, Indian Council of Agriculture Research, on the, and I know he's given so many ideas to the then government to make the policies, particularly in the Sharad Pawar's time, very good experiences. And also he served as a Vice Chancellor of Vishwa Bharati. Vishwa Bharati, he has shape of the, trying to shape of this university. Moreover, he's a very well and I request Professor Shapon Dotto to give your introductory lectures and today's part on that. Thank you, sir.
And Dr. Ghosh, just one line about Shapunda. For others, I think you know, he's the father of golden rice. He's one of the father of golden rice. And we all in India are proud of him because of that contribution. So thank you.
Okay. Thank you for sharing.
Thank you, Professor Shankar Ghosh, also Dr. Shantanu Dasgupta. I think I didn't know that Professor Shankar Ghosh knows so much about me and can talk so much about me. Thank you, Shankar Ghosh, that for your kind words. But I know that your leadership in Castline University made a big change in academic atmosphere. Continuously, you are putting so much of workshop and cutting edge science and new development innovations in your university. And your leadership with the team of your university, you made a very sensational development at the university. We're all proud of your achievements and your leadership at Castline University. We also should thank today Dr. Jennifer Doudna and Emmanuel Carpenter, because of these two ladies got the Nobel Prize and brought these particular genetic scissors or molecular scissors. The today's topic brought all of us today here with Professor Hemant Majumdar, Professor Amit Ghosh, and of course today's keynote speaker is Dr. Anindya Bandyopadhyay, who is basically a zoologist and converted to a plant biotechnologist. I forced him a lot for that anyway, but you would be hearing from him, an excellent worker as well as speaker. As I see also this program today also associated with INSA and NASHI, which is also very nice, and Hemant taking leadership for that, as well as we're all participating in many of those activities in Calcutta. Now, as we'll be talking or listening today, as we heard it before also, genetic or molecular scissors. Basically, in nature, these things happening for many millions of years. And that's the beauty of dynamic nature of biological species. They're continuously evolving, changing, including the host pathogen interaction, evolution, co-evolution. They're continuously changing. One is attacking other is silencing to protect the plant against the invaders, etc., etc. What scientists today is discovering what is happening and why it happened, and that's the way that things are moving. And we are learning and understanding new science and interpreting science in a different way. Even if we take the conventional breeding, plant or animal, then comes the mutation breeding or the later part modern genetic engineering or transgenics, as well as genome-based molecular marker-assisted breeding. Anywhere you go or any method you take, it's a question of time and where it is developed and how it is applied. Basically, all genetic changes taking place with the prediction, with the desirable trait, desirable character, desirable improvement, etc., etc., and with the time of science that we are associated. And today's basically the genetic scissors or genome editing, based on the platforms that developed starting from, say, Mendel, Darwin, or Watson, Crick, and so, and last 20, 30 years, tremendous progress have made. And what we heard from Dodner's talk, the basic platform last 10, 15 years, and that where and how this today's genome editing has taken place. But it also reminds me one thing very interesting. It's not science, but it's basically, to me, it's quite exciting. Rabindranath Tagore's, even his poem, he continuously uses scissors. He changes, remove the words, put a new word, insert a new word, insert something drawing. He developed a new creation out of his poems, out of his correction. His correction also made a new art as well as new expression of poems. So today's molecular breeding or insertion of a targeted gene cassette in a predictable manner so that a trait is developed, or even removing a particular gene, or even inserting something or removing another thing, or separable of another thing like a marker gene. All of these changes, basically, we are looking for a trait improvement, a trait development, or something to remove to make a better trait or better expression of a particular gene. So what Tagore was doing in the poems, in poetry, we see in science also. Basically, we're changing, reshuffling certain alphabets, certain amino acids and coding sequences, and putting certain piece of DNA so that it makes a better expression, better development of a trait. In plants, animals, fungi, or in medicines, or even human gene therapy, now these techniques will have a tremendous impact. And now the new additional information, what is coming up, that is genome safe harbors. A genome safe harbors is certain specific place of a chromosome or in the DNA where the insertion will take place, so that even disruption of genomic DNA will take place, but it would not affect much or would not have any pleiotropic effect. What happened in molecular breeding or conventional breeding, all the time we see this thing happens, but we need to select a material and it takes time. And today's genome editing will do the same thing in a precision manner and at the same time within a shortest time. And Shantanu Dasgupta was mentioning about golden rice. Just I take 1 or 2 minutes for this golden rice. Anindo just sent me today a paper from Pamela Roland. With her, we are working also on bacterial blight XA21 gene insertion in rice. We have been very successful in developing that transgenic rice for bacterial blight resistant. Now, Pam published a paper recently in Nature Communications where he showed, her group showed that golden rice now can be developed by CRISPR-Cas9. Now, golden rice, last 15, 20 years back when we started the work, there is a gene cassette what phytin synthase, phytin desaturase, and lycopene cyclase. These three genes driven by endosperm-specific promoter, the cassette that we developed, or cassette was developed that was inserted in rice, and that made gene expression carotenoids pathway in seed in endosperm. That is golden rice. Even we have been successful in removing the selectable marker gene by putting two vectors so that one carrying the marker gene and one carries the gene of interest. And by simple breeding, we can separate out the marker gene and get the gene of interest, meaning that carotenoids pathway in rice. Now, what Palm Roland's group did, which that work took a lot of time, as well as a lot of selection, etc., was required. But her group today with the CRISPR-Cas9, of course, the same cassette she used, the beta, not lycopene cyclase, the two genes that I mentioned. At the same time, the same promoter were used, but again, the selectable marker gene was in another cassette. And eventually, she developed marker-free golden rice. Now, using the genome sequencing and with the selected sequence information, she could predictably develop golden rice in a simple manner. What I'm saying that today, whatever molecular breeding or conventional breeding developed or given us the strong platform of breeding for new crop development, today's modern CRISPR-Cas9 techniques will accelerate, will make even simpler and more predictable in a simple manner. And that's the beauty of this entire genome editing. And Anindo will be talking on that more details on application is in agriculture and plant and also in other areas. And I'm very excited that this particular technology is rapidly expanding. It's a new dimension, new domain is coming up, so that it will have a tremendous impact. And those who are normally talk anti-science activists, particularly on GM or transgenic, probably will find very difficult to talk against genome editing because it's more nature, it's more natural, perception-wise, scientific-wise, that'll be very difficult for them to talk against, because this science also would be applicable for human genome editing, human therapy, as well as animal sciences. So I'm quite excited to see the future dimension of this particular area. And thank you again, Professor Shankar Ghosh, for bringing all of us together in this platform to talk the most recent modern science and which has a tremendous application. Thank you, Professor Ghosh.
Thank you, Professor Dotto. It's a nice introductory part for today's program. And really, you have given a insight of this technology so that every people or every biological people, they can use this kind of technology where the ATGC is there. There are 4 letters that Rabindranath Tagore has used his pen or pencil to remove the word and verb that makes the in amino acid changing or ATGC changes that gives the different characters. I used to tell in my student or my faculty, the Bengali student should do the PhD in computer science and vice versa. Like a 0, 1, this and that, so that it is a kind of interdisciplinary thoughts has come up in such a way that Rabindranath Tagore has made this kind of part, and really that comes into the in CRISPR-9 technology. If we take it in vice versa, it might be a very good idea that is developed. Thank you once more from University of Kolkata and the Nasi as well. Now we want to go for the main speaker, Anindo. He's a very exciting young scientist. I know him, but I came to more better in this area, when I called Professor Dotto that genetic seizures that got the Nobel Prize on 7th, we are trying to get the web winner on promotion of science based on the Nobel laureate, So I called him, sir, if you give us some kind of talk, this and that. And Professor Dotto suggested that Anindo is doing very well. And now I'm sure Anindo is the leader of leaders in this area. And lots of publication he made in this area, and the Cold Spring Harbor Laboratory, the conferences and different kinds of part where he has gained a good knowledge. And I'm happy that he is also working in association with one of my seniors like Shantanu Dasgupta. And I used to talk Shantanu Dasgupta frequently. And for your information, that we want to create some new department like a genome science, where that students and other can get a very good exciting on that part. So I'm not spending any more time, and I'm waiting for Anindo Bandyopadhyay, and he is a very close association with Professor Dotto even. In these few words, Dr. Anindo, I'm requesting you to give your valuable speech in this program.
Thank you. Let me first try to present my slide. Whether my slides are visible, I'm not sure.
Not yet, not yet.
Some issues are there. Let me try to present now. Yes, please let me know when you can see my slides.
It's coming. It has come.
Okay. Yeah. Okay, let me put it in the presentation mode. Okay, so first, I would like to convey my sincere gratitude to Professor Ghosh, Vice Chancellor, Kolkata University, and Professor Sapam Datta for giving me this opportunity to talk in front of such an august gathering. And I would like to thank all of the dignitaries of Kolkata University and whoever is attending this seminar. So I will directly go to my talk, and then there will be a question answer session, I believe, at the end. So question answer can be taken that time. So I kept the title like Genetic Seizure Nobel Prize in 2020, Science, Application, and Future Perspective. So mostly I have divided my talk in two main areas. First area would be discovery, science behind CRISPR, technology progress. And second part would be different applications of CRISPR. And one case study I will talk about CRISPR-WEV. So before going to my first topic, I'd like to mention that I'm coming from Reliance Industries. And we all know that there are major three areas Reliance work: Geo digital platform, Reliance retail, and Reliance petroleum business. So we are under this petroleum hydrocarbon activities, and we are R&D from that. We are from R&D of hydrocarbon. And this shows the whole campus of Reliance R&D. It is in Mumbai, Navi Mumbai actually. It's called Reliance Corporate Park. And this is our biofuel plant in Jamnagar near the coast of Gujarat. So it's completely automated, state-of-art, world's biggest algal biofuel plant. And this is the lab we are one of the labs in our RCP center. So our major interest is to apply genome editing and other cutting-edge technologies to use in different microorganisms like bacteria, virus, fungi, and especially algae to develop biofuel as well as novel chemicals. And recently we have also started working a little bit on CRISPR application for crops. So I joined a few months back, and I am working with Shantanu and the entire biology team. So let us start the main topic. So discovery and excitement. So in 1987, basically the first time Japanese scientists, they found that bacteria, in the bacterial genome, there is a special area which has repetitive sequences and which has some special characteristics. Later on, 2002 to 2004, in between that period, many scientists worked on that special sequence, and then they termed that area as CRISPR. And many other work related to CRISPR and inserting DNA or inserting any foreign elements, DNA-based foreign elements inside of a genome was going on. But in around 2012 to 2013, Jennifer Doudna and Emmanuel Charpentier, they came up with a new discovery where they showed that these genome editing elements or these CRISPR elements could be taken into the Eppendorf tube, and we can modify it, and we can use it back in the plant cell or animal cell or any kind of living cell for editing a particular area or changing a particular area of genome. So that was really a revolutionary discovery in this whole pathway or in this whole chronology of CRISPR research. And later on, since their discovery, we have seen many scientists are working in this area and many changes happening, and the field is evolving in a lightning speed. So what is the major change? Previously, we used to do gene transfer or modification of some part of genome, but it was random. We could not target a specific area and change a certain area as per our wish. But now with genome editing technology, which is basically CRISPR-Cas9 technology, a very simple and cheap technology, easy to use, where a single protein, this circular one, and this red one, a gRNA, these two components can target a particular region of the DNA. And we can design this gRNA so that it goes to a particular region or target region and do necessary change, which we want. So now what we have after discovery of this genome editing capability of CRISPR-Cas9, we can do targeted change. A certain area of a genome, huge and vast genome, we can target a certain area and we can do a targeted change. So this really brought a lot of excitement in the field. And we considered that the CRISPR craze started from around 2013. And we could see the rapid increase in the funding in that area from private sector, from government, rapid increase in the publication in the area of CRISPR-Cas9, this green line, you can see, hope you can see my cursor. And then we saw that venture capitalists, the more opportunistic capital investment started pouring around this technology significantly, and definitely intellectual property started coming, and many IP applications and IP grants were seen around 2013, 2014, and this bar is continuously increasing with the time. I made this slide just to just to give an idea how this big craze is going on. Recently, Bloomberg has mentioned that this market is going to be $7 billion by 2026. And you can see a lot of work is happening in the Berkeley and Jennifer Doudna and Charpentier's arena, and also Harvard and Broad Institute, MIT. And all these initial tools of CRISPR-Cas9, they're sublicensing to many companies, university institutes, and companies, big companies like GE, Taconic, Horizon, Dow, DuPont, Novartis, Bayer. These kind of companies are really interested behind it. And more than like 1,000 small startup companies are already there working in this area. So institute, academias, as well as industry, everywhere, it's a big buzz and it's a big opportunity for developing something new or bringing some new product. Now, what is the science behind it? Before going to that, this is my favorite slide. I always like to present this slide to any gathering, that whoever has eaten yogurt from any shopping mall, they have already consumed a lot of CRISPR-Cas9. So last 20 years or 15 years, we are eating CRISPR-Cas9 very frequently through yogurt, because most of these yogurt or cheese companies, they use bacterial strain for yogurt production. Now they found there are certain bacterial strains which are basically resistant to fudge virus, the natural enemy of bacteria. What happens, this rectangular shaped pink box is a bacterial cell, and this one is, whether my cursor is visible?
Yes, visible. Alindo, everything's fine.
Okay, so these fudge virus, they basically introduce their genome inside of the bacteria when they attack bacteria. Now bacteria get sick. It cannot do anything, but one thing it does, it cuts that part of genome and internalize it into its own genome. So in the CRISPR region. So later on, when that same group of viruses attacks that bacteria, they use that memory of previous attack, and they develop this CRISPR-Cas9 tool to resist the newly invading viruses. So this CRISPR-Cas9 is nothing but bacterial, adaptive, or memory-dependent immune system. So Danisco is one of the biggest companies. Later on, got acquired by DuPont, and they basically use these bacteria which are resistant to these fudge viruses. Later on, with different research, they realized those resistance bacteria are having CRISPR arrays in their genome, and they have a memory so that they can resist viral attack. So they started selecting those bacteria more and more, using those bacteria and selling those bacteria to the yogurt companies. And therefore, most of the yogurt companies or cheese companies in today's world use those bacteria for cheese or yogurt production, which has CRISPR array in it. So bottom line is, last 10, 15 years, we have eaten a lot of CRISPR. Now, there are two types of CRISPR system mainly available in the world, in the bacterial world. So one is class 1 and class 2. Class 1 has a single gRNA and many Cas proteins together, and it is very difficult to handle this class for use of genome editing. But class 2, which has a single Cas protein and one gRNA, is easier to handle, and people are using this class 2, different types of class 2 CRISPR-Cas9 system for genome editing. Now, the bottom line is CRISPR-Cas9 is a simple, very easy to use system which requires one filament, which is a gRNA, and a protein, which is a Cas protein. So what it does, you can design your gRNA and you can take a Cas protein, fuse them, they will make a complex. Now, gRNA will take this whole complex to a particular region of the genome, which you will decide, and accordingly you will, or scientists will design that gRNA area, and it will give a double-strand break. When this double-strand break happens, cell cannot stay in this way. They try to repair. Intrinsically, they try to repair, and while repairing, some error happens, some mutation arises. So these mutations basically edit that part of the genome, and that gene cannot work anymore where this mutation happens. Alternatively, you can use that same Cas9 gRNA and send it to a particular region of a genome. You get the double-strand break, and then you supply artificial gene from outside or a gene taken from some other organism or from that same organism, and you supply that in front of the cut region. That supplied gene can get swapped in by homology-directed repair. Similarly, you can use multiple gRNA so that in multiple areas of a gene. So it's a very flexible, easy to modify, and easy to use system, and which can be used in any kind of cells where genome is there. Not only that, gradually people started observing that you take that Cas protein, one of the component of CRISPR-Cas9, and attach something of your interest, and you can do something else. Like epigenetic regulation, the regulation beyond gene, this methylation mediated or acetylation mediated, there you can fuse epigenetic regulator with your Cas9, and you can modify the epigenetic status of a cell or of a genome. Similarly, if you hook activator or repressor with the Cas9, then your activator or repressor can be used to target a particular gene and activate or repress that gene. So different modifications started coming up very fast since the discovery of the CRISPR-Cas9 in 2013. Also, people started showing that you can change the gRNA in such a way so that it can attach with fluorescent molecule, and then you make this D-Cas9 inactive, so which is called dead Cas9. So you send a dead Cas9 with a gRNA hooked with a fluorescent molecule, and under microscope you can see a certain area of the genome in a colorful way. So similarly, different other pull-down techniques which people used to use in molecular biology randomly became much more easier and faster by application of genome editing. So in this diagram, I am summarizing the whole thing that CRISPR-Cas9 not only does double-strand break, like left top, it can do double-strand break, we already know. It can also do gene repression. It can also do gene activation. You can tag a certain area of a genome. You can purify a certain genomic loci. You can image by attaching a fluorescent molecule with the Cas9. You can do a genomoid screening, or you can even do a nick. So CRISPR-Cas9 has two lobes for cutting double strand. Now, if you inactivate one lobe, it will cut only one strand. So it can give a small nick, a single strand cutting characteristics you can render on CRISPR-Cas9. So this simple molecular tool can do various molecular activities which are very important for molecular biologists and for multiple research and product development activities. So what happened since then? So from 2013 to 2014, 15, 16 until 17, people use this CRISPR-Cas9 for and modifying this CRISPR-Cas9 system for various activities already I have mentioned in a molecular level. Later on, many labs around the globe started working on it. And every day, some new paper comes out, a new technology comes out based on CRISPR-Cas9. So I will briefly touch some of the very interesting ones or very important ones which came up in recent days. I thought it would be great to mention those while talking about CRISPR-Cas9. The first one is base editing. We know that single nucleotide polymorphism are responsible for many characteristics. There are many human genes. If a single nucleotide gets changed, then certain disease happens. Similarly, in plant, there are many genes. If single nucleotide is changed, then some beneficial characteristics we can get out of that gene. So far, CRISPR-Cas9 was used to do double-strand break and bringing mutation in a gene and stop that gene activity. But later on, people realized, especially from Harvard University, David Liu and team, they first showed that you can take a CRISPR-Cas9 system, you inactivate the Cas9 protein. So CRISPR-Cas9 system will go and sit in a certain area of a genome as per your choice, but will not cut. Right? Now you fuse a deaminase along with this Cas9, then the deaminase will do one base change only. So you can change a single nucleotide polymorphism. So one single nucleotide can be replaced by another nucleotide. So there are three types of genome editing later on people realized: CBE, ABE, cytosine-based, adenine-based. So C to T conversion can be done, or A to G conversion can be done by these two. And recently, people have found that certain modification, if you attach one adder, adenosine deaminase acting on RNA, you can change this base editing system in such a way so that it can change RNA, a single base in the RNA even. So genome editing by CRISPR-Cas9 now went to a different level where not cutting a DNA or cutting a gene and stopping its activities. Now you can change a single base even by these very powerful modified CRISPR-Cas9. Later on, one more modification came up, which is called CRISPR-X. It came out last year by Michael Basic from Stanford University, where they modify the gRNA and they attach one MS2 trap gRNA where multiple cytidine deaminase can be hooked. So this green part is your Cas9, and these are the gRNA. And now you attach a lobe here, which is MS2 trap, and multiple adenosine deaminase you can hook there. So those adenosine deaminase, what they will do, they will change single nucleotide, many single nucleotides in a certain area. So you can get random mutations, a large-scale mutagenesis, a certain area of your choice. So it has many applications, and it's smaller in size, pretty helpful in different kind of research and product development activities. One more massive change came up recently, which is known as CRISPR or retron technology, again, path-breaking modification of CRISPR technology, where retrons are natural DNA elements which code for a reverse transcriptase as well as a template on which the reverse transcriptase act to create a multi-copy single-stranded DNA. Now, you take this retron, attach with a gRNA and CRISPR system so that you can supply your gene of interest very easily inside of a cell. And this retron RT-mediated CRISPR system, which is named as CRISPR, can very easily insert your gene of interest inside of your target site. So this is also one thing. Later on, people came up with different other fusions. Great amount of protein engineering started happening, where one of those is evolve-R, where what happened, people took CRISPR-Cas9 system and attached one error-prone polymerase with the Cas9. Now you send that whole system in the inside of the plant cell. What it will do, CRISPR-Cas9 will give a nick because you are sending nick version of CRISPR-Cas9, where one lobe of Cas9 is inactivated so that it cannot cut double strand. It will give a single strand cut, a nick. And so first this CRISPR-Cas9 will bind. It will give a nick. After the nicking, error-prone polymerase will bind, and it will try to repair that area. We will not wait for cell to repair it. We will repair it by error-prone pol. So it will give mutations in that nicked area. Then the whole thing will be dissociated. So now you send many gRNAs. Then what will happen? This host system will go to many places of the genome and will cut those areas. And error-prone pol will make mutation in those areas. So in a tunable window, a large chunk of chromosome, you can make different kind of mutations for various purposes. So this is called evolve-R technology, a very powerful technology recently came out. And again, from Stanford, and people are applying quite well. In the plant system, those who work in the plant science, they know that we do gene transfer in plant by Vir genes or agrobacterium-mediated transformation. So these Vir genes attaches a certain area of agrobacterium genome, which is called right border. So if you take a right border sequence, attach your gene of interest, and then with Cas9, you bind your Vir gene, then this right border sequence and your gene of interest will attach with the Vir. Then this whole system, CRISPR-Cas9 system, is sent to the genome. Now what will happen? CRISPR-Cas9 will do its job. It will cut, it will give a double-strand break. But now it is attached with a Vir gene and your gene of interest. So that will come in the near of the cut site, and homology-directed repair will insert that gene of interest inside of the genome. Why these insertions, modern ways, or CRISPR-Cas9 mediated modified insertions are so important? Because we know that homology-directed repair in plant is very, very low. It's unlike animal or human cell lines. It's very low. It happens very rarely because supply of the donor or supply of your gene of interest in front of the cut site is a big challenge. People tried in many ways, in a viral method, in other methods, but recently this CRISPR-Cas9 mediated Vir-D2 fusion shows a real promise that we can change a certain area or we can insert a certain gene of interest in the CRISPR-Cas9 cut site in a very easy way. So similarly, Feng Zhang's group in MIT, they found that there are certain kind of CRISPR in bacteria which can attach with jumping genes or transposes and can facilitate genome insertion in much higher level. And the last and most important in today's world, the modifications and new version of CRISPR-Cas9, which people are using, I must mention, is known as prime editing, where gRNA is not used anymore. A big PEG RNA is used. In that PEG RNA, you attach your gene of interest. So your gRNA will go and bind in a certain area. It will carry the Cas9 with it to that area of the genome. Now you fuse a reverse transcriptase in that Cas9, then what will happen? You send your gRNA attached to its gene of interest, and Cas9 will give a cut in certain area of your target area, and then reverse transcriptase will copy the edit template which you already sent attaching with the gRNA, and your gene of interest or will get copied inside of the genome. So insertion, which is very important for genome editing, is becoming more targeted, more regulated, and this has huge impact in medicine, in disease research, in disease mitigation, and also huge applications in plant science and in agriculture. Last, one thing I must mention, that artificial intelligence is also playing a big role along with CRISPR-Cas9, where people are coming up with new algorithms like Forecast, Sprout, or in Delphi, where a scientist can predict if I cut a portion of a genome by CRISPR-Cas9, and if I allow cell to repair it, what kind of repair that cell will do. Previously, even last to last year, people used to think, scientists used to think, this cellular repair mechanism is random. But recently, people have found out that no, it's not random. There is a fashion which cell's intrinsic machinery uses, so that these changes which cell will do, we don't know what it will do. But yes, by these algorithms, we can predict even what kind of change cell will do. So I was mentioning, and Dr. Datta and other speakers were also mentioning, that this field is moving in a lightning speed, and huge amount of concentration from government, from academia, from industry are behind this field. So if we think, so what is the bottom line, or what is the application of all of these technological or technical development or technical improvement of genome editing. So genome editing by CRISPR, especially many modifications. Every other week you will, if you follow this field, you will see every other week there is a new modification and a new benefit. So what is happening in actual field? What is its application? So here I kept three things. We are interested in algae. So in algae, you can very easily send this CRISPR-Cas9 by electroporation. In human, you can send this CRISPR-Cas9 tool by polymer or peptide or viral vector or liposome-mediated pathways. Or even if you can microinject in the ovum, in the germline, but in case of plant, microinjection or these easy ways are not available. In case of plant, what we do, we still do agrobacterium or ballistic-mediated transformation, but there are new things coming up involving CRISPR-Cas9 as well. And all these transfer techniques helping us sending the CRISPR-Cas9 tool inside of the cell, and we can apply this CRISPR-Cas9 for our benefits in many areas. In disease resistance or disease research, what we have seen in cancer therapy, this technology is using greatly. Cancer immunotherapy, GE, genome-edited immuno T cells are very common nowadays, and already many products are in human trial. One of that, like esophageal cancer, PD-1 gene, people have knocked out, so that cell are not cancerous anymore, and it's in human trial in US. Similarly, you can knock out a miscarriage gene, and you can improve fertility of human body. So it can. Many other in vitro gametogenesis studies, people are using CRISPR-Cas9 for myriad purposes. Different genetic diseases, gene-mediated genetic disease such as genetic blindness, cystic fibrosis, Huntington's disease, beta thalassemia, etc. So, or DMD, hotspot removal, etc. People are using genome editing, and few are in pipeline for regulatory approval. Similarly, infectious disease, lot of happenings. One very controversial happening, in China, what happened, one scientist basically did embryonic genome editing. It was too early, although he is in jail. Unfortunately, he hasn't taken any regulatory approval or anything. But what he did, he took genome editing tool and changed the embryo of HIV positive parents. And they could successfully give birth of babies which are completely HIV negative. Although there are many things happening in this area, but a lot of regulatory hurdles are there. People have to obey, otherwise anything can happen anywhere. It's a very powerful technique. We have to be very careful. Also, organ transplantation. We all know that many organs like kidney or liver or lung are in high demand. So we really need a powerful tool for organ transplantation. So what people are doing, infusing human stem cell injected in pig by CRISPR, and they are developing hybrid organs. Similarly, in drug discovery, in many steps, animal model creation and genome editing or CRISPR is in great use. Different diagnoses, including this COVID-19 diagnosis. We have heard about Feluda. Recently, one of our CSIR scientists had developed. Previously, Broad Institute has developed, and we have published a paper in this area, and we have shown different CRISPR-mediated method of genome editing to identify diseases, especially viral diseases like Ebola, Zika, HPV, Corona, etc. We can really use this tool to very fast diagnosis of the diseases. Similarly, animal models are in great use. These are the monkeys, rhesus monkeys in China, where circadian rhythm gene BMAL1 is knocked off. And what is happening? A lot of new findings, new discoveries came up related to insomnia disease, sleeplessness. Similarly, muscle-related discoveries in these genome-edited dogs or swine flu resistant discovery in these genome-edited pigs. In many things are happening in these animal model systems. not restricted to the disease or animal model. We know this pet breeding is also taking help of genome editing. These Dalmatian breeds are known for kidney stones. So one group of scientists in US, they just mitigated this problem by editing one single gene so that Dalmatian breed are not having any kidney stone anymore. Similarly, faster horse bred with much more muscle, healthier fish, dehorned cows, and modified pests for gene drive can be made and in production in different areas of the globe, in different labs by genome editing. So it's a great field. And in Reliance, we are extremely interested in using this CRISPR technology for industrial biotech. What we are doing, we are taking organisms, especially algae, fungi, plant cells, yeast, bacteria, etc., and we are changing them by CRISPR-Cas9 so that they can produce better. Now, we develop those organisms, modified organisms, then we develop process, and then we do scale up and piloting so that generate different kind of very important chemicals used in pharmaceutical industries, used in cosmetic industries, used in feed industry, used in biofuel related activities. So many other activities, very important chemicals can be produced by enriching your cell lines by genome editing or CRISPR-Cas9. As I mentioned, and I showed this picture previously, this is our state-of-art, world's largest algal biofuel plant in Gujarat, where we are using this genome editing tool, especially CRISPR, to modify microalgae so that we can produce a good quality and good quantity of bulk chemicals, biofuels, lipid, biogas, and pigments by these algal cultures. These are all algal ponds. We are also doing reactor-based algae development and production of important chemicals from algae. Similarly, agriculture, there is revolutionary change in agriculture. The traditional breeding process in agriculture takes 8 to 10 years. Mutation breeding also takes 8 to 10 years. But by genome editing, within 1 to 2 years, you can develop a product. Then most of the countries, many countries in the world, they are considering genome editing as a traditionally bred product. So regulatory hurdle is much, much lesser. And you can develop a product very fast, where traditional breeding takes 8 to 10 years. You can go to the market with your product, or you can make a product in public sector, give to the farmers within 2 to 3 years, maximum 4 to 6 years. So this is really a path-breaking tool, basically changes the way we think science in various fields. Now, for the plant or agriculture biology, those who are working in the agriculture field, we all know this equation. This is known as the equation for, or breeder's equation, or equation for genetic gain. So I am not going in the very detail of this equation, but just I will try to bring your attention in L and C. So breeding cycle is L and cost is C. If you can reduce breeding cycle, time of breeding cycle in factor of 2, you can basically double the genetic gain and you can reduce the cost significantly. So for industrial activities, even for academic activities, if we can really reduce the breeding time and then we get double genetic gain. Also, we could reduce the cost significantly. So this is really helpful for farmers, helpful for production and other players present in the chain or supply chain of this whole field. So where CRISPR basically fit in agriculture. So we know in agriculture, design crop products people are producing since long back, either by traditional breeding, where you are mixing two genomes in an unknown fashion, and randomly you are mixing and you are waiting for the progenies and testing those progenies and finally finding out a beneficial progeny and selecting that for ages. Or you do mutational breeding by chemical mutations or by radiation, or you do GM, or you do genome editing. So in this whole blueprint of plant breeding, where this genome editing fits, basically, if we see this genome editing, so it can rapidly help in functional validation, it can help in diverse allele creation, and rapid trait integration towards designer crop product development. So yes, we can really apply this technology to expedite breeding and produce product in a much faster, much cheaper, and in a much easier way. So at the end of these applications of CRISPR, I would like to mention one thing. All of the agriculture biologists and other biologists, they know that haploid breeding is very important for commercial breeding activities or even public breeding activities. But there are many bottom lines in today's plant world. Different elite lines or the good lines which we really want to use for production of our crop, rice, wheat, or corn, or whatever. So elite line transformation or sending your genome editing tool or any tool or gene of interest is a very challenging. So elite line transformation is still a bottleneck. Many elite lines are recalcitrant to transformation methods. Delivery of CRISPR tool is a big issue. And traditional integration, I mentioned, it takes 8 to 10 years. So it's very lengthy, time-taking, labor-intensive, and costly. Recently, there was a publication last year by Kellier and Chu, came out in Nature Biotech, where they have used this CRISPR technology so intelligently, and they fused with haploid breeding. So you can bypass transformation, no need of transformation. First, you develop a CRISPR-Cas9 containing plant, use a very simple, easily transformable line, and you mutate its CNH3 or any other gene so that it is haploid inducing. Now you take the pollen, just simple pollen from that plant, and you transform, you pollinate any of your elite lines. And end of the day, what will happen? That elite line will be edited. In one shot, you will get a haploid elite line. I'm not going in detail why haploid is so important. Those who are from this field, they know that haploid is really important for product development. So you can do haploid and genome editing simultaneously in single shot. What it's doing, you are taking the pollen. That pollen is having a genome editing tool, that pollen chromosome actually having genome editing tool, and it has a mutation in its NH3 gene. Now, when that pollen fuses with a normal or elite line egg, what happens? From the pollen chromosome, genome editing tool cuts the egg chromosome and do the genome editing. Now, this pollen chromosome has haploid-inducing mutation. So the pollen chromosome will fall off in the zygote. When zygote is formed, this pollen chromosome will fall off. What you will end up? You will end up with a zygote which has only one chromosome, the mother chromosome, the maternal chromosome, the chromosome from an egg, but it's already have an editing done by pollen chromosome previously. So this edited line don't have any more the male part. So male part is fall off due to CNH3 mutation. And while falling off, it took away the genome editing tool. So end of the day, you are finding a clean haploid without any genome editing tool. And that haploid is already edited, edited in the gene of your choice. And then you double it, you get a double haploid. So within one generation, you get a single fixed line double haploid with edit, without transformation, without any CRISPR-Cas9 tool. So these kind of path-breaking technologies or path-breaking applications can be done by genome editing. So already people have made many products. This product, this non-browning mushroom, we know. If you cut a mushroom or an apple and leave it on your desk, you will see it will be black due to one particular gene, polyphenol oxidase. Now, scientists from Pennsylvania State University, they cut that polyphenol oxidase gene in genome and they produce a mushroom which will not get blackened for a long time. Similarly, hop-free beer, in beer industry, in brewing industry, hop is used to give a particular flavor and froth in the beer. So you can just change 2 genes in barley. Then what happens? You don't need this hop anymore. Hop is costly and it requires a lot of water. So you can use time, money, and also you can save a lot of water. Similarly, spicy tomato, waxy corn, decaffeinated coffee, gluten-free wheat. So many products are coming out. And interestingly, most of the part of the globe, these are considered as a single base change. So naturally, we all know that all organisms evolve. And if you see the plant, if you sequence a genome today, and if you sequence it 1 year later, you will see a lot of mutations. Naturally happened in the genome and gave rise a new trait. So you are just mimicking those natural CRISPR-Cas9 tool. CRISPR-Cas9 tool going inside of the cell, making a mutation coming out is just like a simple mutation. And no foreign element is sticking there or staying there. And later on, when you make a product, it's just similar to natural mutation products. So most of the part of the world, like US, Canada, and many countries in Latin America, they considered these genome editing mediated knockouts are considered as naturally bred. Similarly, in Australia and Japan, they have also decided. Unfortunately, European Union gave a verdict that any modern mutagenesis technique will come under GMO. But a lot of countries like Germany, UK, Spain, they have come up with their own, they are trying to come up with their own regulatory activities. And they requested European Union to reconsider because genome editing is a very powerful tool and it should be, and European farmers should get leverage this technology for their development. Unfortunately, in India and China, it's not anything negative, but it's still under discussion. So no final discussion, so no final decision came out in India for genome-edited product to be used in for public consumption or for the farmers' field. So this is, I tried to give idea how genome editing is used in different organisms, especially in agriculture. And I will take just 5 more minutes to briefly describe how it works. I work personally in these activities with University of Sheffield and International Rice Research Institute, where we took a rice plant. We know in any plant, there are small pores, which are known as stomates or stomatas. The pores are useful because carbon dioxide get inside of the cell. By these stomata, oxygen and water come out. So these are really important. If you can reduce number of stomates, then what happens? More water will stay inside of a plant body. So the plant, those who cannot grow in the dry or arid area, where drought-prone areas, there we can send a rice plant or grow a rice plant because it has reduced stomata. It can hold more water inside of its body. So we used CRISPR-Cas9 and CRISPR-CPF1, both type of CRISPR system. And in the pathway of stomata development, we choose few genes. We knock one of the regulatory gene known as EPFL9 in the exon 1 using both CPF1 and Cas9 in 2 separate experiments. So later on, we could see editing has happened by application of genome editing tool. And after few generations, the genome editing tool, CRISPR-Cas9, is segregated out. And this is the top picture shows the blue yellow stars. These are basically different stomates in a natural rice plant. By application of genome editing, we could change in few stomates 8 times reduction we can do in the stomatal numbers. And we could see a huge improvement of water use efficiency. And we can grow those plants grow better in drought and arid conditions. Later on, we also found we did off-target analysis, and we could see in this particular experiment we did not have any off-target. And later on, it was followed up. Many publications came up later on with this, and more advancement in this area took place. And many publications came out. This was our initial paper, where we show CRISPR-Cas9 and CPF1 mediated a change in stomatal number. And then in cytopathology, further advancement was shown. And Virginia Tech University requested us to give these news in their website. So a very simple way, we just change a single gene and we can transform rice, which requires a lot of water, cannot grow in a drought area or dry area. We can use that for different purposes. So that's it. Future perspective, if we see, where are we? Definitely we need continuous improvement. We have to do protein engineering and make this CRISPR-Cas9 system more better. We have to address the bottlenecks like delivery problem, immune response, molecular protection, etc. And we have to develop application pipeline. Whatever we do in the lab, end of the day, we have to use it for society, either through government channel or through academic channel or through industry channel. We have to develop this application pipeline. We have to merge artificial intelligence and data mining with it. And also, we have to have manufacturing infrastructure and scale up to bring this whole thing in reality. So this is our labs in RCP in Bombay. And this is our team, biology team. I would like to thank Shantanu and the whole team. So I joined recently, and this is a very fascinating team working in the area of biofuel, synthetic biology, and there are many engineers also involved, chemical engineers, for scaling up of biofuel, chemical production, and application of CRISPR for synthetic biology. So thank you for attending, and thank you again, University of Kolkata and Vice Chancellor, for inviting me to give this talk. Thank you.
Thank you, Dr. Anindo. It's a nice deliberation, and it is a really a scale up of the entire process of getting the Nobel Prize in this area. You have mentioned really from the beginning to the future scope, and giving the point, every point, how it develops very nicely. Within a short time, the CRISPR-9, CRISPR-Cas9 technology has given a wide application where ATGC is there, where protein is there, where the phenotypic expression is there. We can use this technology, right? So in near future, any biologist, not in biologist, I can tell you, the chemist people and the physics people and artificial intelligence people can make a interdisciplinary approach, which can give a very, very nice tool. It is a very nice tool that we can get more efficiently, and he has explained very nicely. Everybody can understand and pointed out a very nice deliberation with these few words. I really appreciate your effort, and we hope we can get more expectation from you, from your leadership in Reliance in India, and your team, as well as the Shantanu Dasgupta also is associated with that. And we are expecting that you, the Bengali people, should lead the CRISPR-Cas9 in India and a global platform. We will be happy on that area. Very nice talk. And thank you once again, Anindo, from university as well as the NACI program. Now, I want to request mm, mm, Professor Amit Ghosh, who is a very renowned person in India and abroad. He has basically physics man. We know that he is a physics people now interested to go for in biology and work a lot in work a lot in not in India, in abroad, and he developed a nice part in India. And if we, Professor Amit Ghosh is a, I have associated with him long back. And when I was in Northeast, particularly, just I am sharing some of my experience. When I was in the Northeast in Assam University, Silchar, we have arranged a one NASI program for promotion of science, and the expert from the NASI people from Calcutta, they went in the Silchar and given a talk in variety in different colleges, and students are so excited with their promotion of science lecture. Still now, I am getting call from the principal of some colleges, sir, ওই রকম program কিন্তু আমাদের বারাক ভ্যালিতে আর কোনোদিন হয়নি. So you see that was a December 6, 7, 8, and the Professor Amit Ghosh has given a wide lecture in that area. And I just want to share that he is a expert in all academics in a position of executive members, and given drive on the particularly in biotechnology, molecular biology, in special reference to microbiology. He is developed a microbial repository in India, particularly, and now working as a NASI scientist in NICET, Kolkata. And he was the director of MTech Chandigarh, and he is one of the instrumentation in biotechnology revolution and developing the different kind of institute in India, particularly in Gujarat also. And now we want to say sometime, Dada of Dadas. What are the scientists that are working over here? He is the expert of experts. We want to say like that in our particular view. So I just with a few words, I request Professor Amit Ghosh to give a sum up and who's the areas of this Nobel laureate as coming in such a way, and give some views. After your talk, we can make a question answer on that part. Professor Ghosh, kindly.
Thank you very much, Honorable Vice Chancellor. Since most of the people listening to this talk, I believe, are Bengalis, so I will start with a Bengali proverb. বাংলায় বলে যে আগে থেকে যদি খুব সুন্দর বলা হয় কোনো মহিলাকে, তাহলে যারা কোণে দেখতে যান তারা ডিসঅ্যাপয়েন্টেড হন। With Shankar's effusive, praises about me, have nothing of it, but I am very grateful to him because he is in a way, he's a student of mine, and students always say good things about their professors. I don't know about now, but at least they used to do it. Now, we just had a very brilliant and very informative talk by Dr. Bandyopadhyay. I understand he worked in Jennifer Doudna's laboratory, so probably he had a ringside view of how this whole thing happens and how science is done in some of the very great laboratories in the country. But he sort of, for obvious reasons, shortage of time, I believe, shot through the introductory part of it. What I intend to do is to tell us about how this whole field unfolded. And at the end of my comments, I would also like to say a few words about what is my perception, or it's not my original idea, but idea of some people, of how science is done and where it takes us to. So I first begin by showing you a picture of a person who is, you may know this person. His name is Virginius Sixsmith. And this gentleman, you may have heard, many of you may have heard, was a close contender for the Nobel Prize this year. Now, what happened was that he independently discovered and found out what Jennifer Doudna and Emmanuel Charpentier did. But this is one of the strange things in science, that his paper, when he sent it to Cell, was rejected. Cell didn't send it out from the editorial rejection. And then it went to PNAs. One of the reviewers was Art Landy, a very well-known man in lambda biology. Either Landy didn't understand the import of it, or for whatever reason, his paper got published 2 months after Doudna Charpentier's Zinc et al. Paper came out in Science, even though his paper, the paper of his group, was submitted 2 months before a paper by Doudna and Charpentier came into being. So it's a question of priority that is always important. And if I may digress here a little, there's a very interesting book called Politics of Excellence. It talks about the level of politics that happens even at the Nobel Prize level. But I'm not suggesting that it happened here. I'm just stating a fact which came out all in the public domain. Now, how did this thing all happen? We all know about this gentleman, but how this whole business of CRISPR-Cas methodology came into being. Now, I go to this gentleman. His name is Francisco Mojica. Now, he was a graduate student in a place called University of Alicante in Spain. Now, he went to the university there and wanted to do research, and his guide gave him a problem, which his guide discovered. The problem is like this, that this place is very close to the sea, so a lot of halophilic organisms were there. So he found that a particular halophilic organism, if you can change the salt concentration in the growth medium, the restriction cleavage pattern changes. So he asked this Filippo Mojica, to investigate that problem. And when Mojica started investigating, started looking at the fragments that got generated, he found that there's a kind of repeats there, which are separated by spacers. He was intrigued by it, but of course he didn't understand it. But anyway, he sent out the paper, first to Nature, all top journals rejected. So finally, after about a year, his paper got published in Journal of Evolutionary Biology. So the message here is that not necessarily these great big journals and their reviewers can always understand or look into the future and truly understand the import of a work. And then what happened was, when he got very intrigued, and so he really spent his time working on it, and then discovered that another gentleman, his name is Atsuo Nakota, he's in Japan, found similar kind of sequence in E. coli. But they're not interested. They were not intrigued by it. They just found it. So what happened was, Mojica started investigating, and about I'm really condensing it. If you want to know the history, everything is out there. You can read it. He made an analysis of 4,400, spending weekends and all of that, and eventually discovered that such sequences are very widely spaced. And this, of course, is taken out from a very controversial article by Eric Lander. But what it shows, basically, that starting from Alicante, where the where this Mojica worked, it eventually went all over the place. And Dr. Bandyopadhyay mentioned about Danisco. The scientist there was actually working on a particular kind of a bug. And there's a whole lot of story. And incidentally, I may mention here, the CRISPR, this particular term was also coined by Mojica along with some Dutch scientists. Now, what happened was that when he sort of spent all his time working on it and showed that this is over there and kept on it, worked on it, but finally, somewhere along the line, he sort of was overtaken by more, I would say, more powerful work by Doudna, Carpentier. Then the application part came from Church's laboratory at Harvard, who showed the actual application of it. And you heard that one Chinese scientist is in jail because he tried to go forward. Now, what I want to tell you that this whole history gives us some message. First of all, in science, there's a distinction between discovery, invention, and innovation. Discovery, you can understand, discover. You are uncovering something. And that is what happened with the work of Mojica and this Japanese scientist. And then this, the names are not coming to my mind, the French scientists, the people who are in that company, Danisco. So for them, it was a discovery. The invention is creating something totally new out of one's ideas, not necessarily one's own ideas. A very good example will be cell phone. Everybody knew about electromagnetic radiation, but cell phones, they didn't think about it. It was created. It's the invention of man's mind. And this is where the people who are, like people like George Church and others have done. And then innovation. If I am permitted to say so, with due apologies, we in India are good at it. There are 12,000 patents on CRISPR-Cas. So many laboratories are working on it. You will find everywhere we are kind of also running. Some new thing comes out in the West. A simple thing, maybe discovery of a repeat sequence like Mojica and others did, and then pursued it. But the original idea, the original observation comes in the West. We innovate. So many laboratories in India are working on this technology, doing very important things. Innovations are not unimportant, but there is a difference between discovery and invention and innovation. Now, I will not take a lot of time. I just want to say what are the messages that we get. The first message is priority is, of course, important, but it's not necessarily that the paper has to be in really very high impact factor journal. Cell is something like 50 or something, but Cell missed it. Cell missed it because erudite people in Cell couldn't understand. Second point is, it all depends on where are you. I mean, when Art Landy looks at Lithuania never had a molecular Nobel laureate. We knew about Lithuania because they were making very good restriction endonuclease. In fact, this is the man, Virginius Sixins. He was a person, and if you look at the catalog of Sigma, they were selling this restriction endonucleases. So here is an unknown man coming from nowhere versus some top-notch places in the world where is a very good tradition. Obviously, when a publication or something very new comes from such a laboratory, we have more trust. You cannot blame. Like if you really publish something very great from India, the problem is the reviewer may think with the corruption level almost at 100%, are these guys telling lies? So they will rather wait to see if there is a confirmation. Situation may not be as bad for East European countries or Lithuania and other places, but it does matter. And that is why you'll find sometime many of our scientists' best works are when they're in the West. After they come back, there's no diminution of their intelligence, but somehow that level is not reached. Second thing that comes out is a dedication. I mean, look at this man. I mean, look at anybody. I mean, I think Dr. Bandyopadhyay would be the right person to tell you about about how science is done in these laboratories. The intense work. You look at a problem, you literally sleep and think and come out of it. So that is something which is very, very important. Another point is, with passing days, I think Nobel Committee will find it very difficult to find out whom to give the Nobel Prize. Because if you look at the subject we're talking, you know, but for some wet lab experiments, the whole idea, when Mojika looked at 4,400, he used bioinformatics tools. So the big data becomes extremely important. And if you are not conversant with that, we don't have the wherewithals to really do that. No matter how well we do, we'll always come out, I don't want to say loser, but come out not at the top of the race. So any paper today will, if it's, even in physics, you know, when Higgs predicted Higgs boson, he was not given Nobel Prize till it was the particle which gives weight to other particles, boson, was discovered, and then he got the Nobel Prize. So the question is, but if you look at that paper, like you see that any paper today comes, you have huge number of names, and all the people who are listed, they have made some contribution. It's not that they're just there by default. So the question is, another thing, unless we have these, the days when an individual lone scientist working in, say, X university, no matter how brilliant, is unlikely to find a place in the sun. So I don't know how to solve these problems. And that's why even in India, if you look at the awards, rewards, prizes, you find is all centered around some so-called important institutes. Important I'm using in a particular manner. But what gave India a place in the sun? All experiment, all work was done in the university system. But today, the university systems, you know, the grant comes and it's distributed among 40 people. Some of them are very good, but some of them are not so good. But you have to distribute it in a democratic manner, otherwise you have a problem. And then these new institutes get all the money. And so this is a skewed system. But again, I feel that universities are the places where these things should happen, because when a particular student comes out to do research in a national laboratory, he's already 20, 24 years old. His basic idea, basic idea about what science is all about happens in the university system itself, and we are forgetting that. This is my personal view. And one final point I would like to make. It's very controversial though, but I don't care because I have nothing to lose. It's a societal value. That's why you find that everybody wants to be an engineer. I mean, sorry, an IAS officer, an IPS officer. You know, some of the people who are present here are very good scientists. I know for sure. But if such a person goes to the airport, I have seen Professor Ajay Shud, who is an FRS, has made fundamental contribution in physics, standing in the queue, and a Thanadar, I say an IPS officer, comes with some retinue, jumps the queue and goes in and gets a lot of salute. Now, I am a young person. I look at it, I decide, forget about what I can do in future. I would rather be an IAS officer. And that's why you have DGP Police of West Bengal. He's a mechanical engineer from IIT, but he chose to become an IPS officer. The society also gives an information. And as a result of that, many students are coming to research by default. And this is not a good sign. And I'm saying this because I think it will be is being attended by some students and educationists. I would like to tell to the students that the ultimate pleasure that you get, like you look at Dr. Bandyopadhyay. He's a vice president at such a young age of a major company in the world. I mean, so this is a path which gives you glory, which gives you power, it gives you also money. I'm sure Professor Bandyopadhyay is quite rich. So it's not something that you have to always go sit for these common entrance examinations and do all kinds of things. This is, I would like to stop here. This is not strictly connected with the subject matter, but the lesson you get if you look at the history of this whole development is something that came out so loud and clear. So that's why I thought I'll mention some of these things. I thank you once again, Shankar, for inviting me and sharing some of my thoughts. Thank you.
Thank you, Professor Amit Ghosh. It's a nice part, though it is a scientific area, but we cannot ignore our societal benefit or scientific arena. That's a true sense that research is not for all. Always we want to tell, and students should get some kind of motivation and interest on the research so that they can give or explain something to the society by making on that part. It's a very nice deliberation, and all the research scholars, students, and viewers also can recognize this kind of part is happening throughout the globe, not in our country, and everywhere these things is happening. So might be careful in every time and respect our science and society so that we can able to get a good impact on the society, because we are all are for the society and the science. So that's a very good idea. And once again, I thanks Professor Amit Ghosh for giving this kind of very elaborating idea about the Nobel Prize and this and that. And so in physics, if I feel that Bose has not awarded to Nobel Prize, but we can recognize that throughout the world, 50% working in the physics based on the boson, and so many people are got the Nobel Prize based on the boson particles, but Bose is not was not awarded to Nobel Prize. All we know this kind of part but though we have to get this kind of information anyway. Now the question session. Some question has already raised by the participant. Professor Ashok Babu, please continue the question session with the expert. Ashok Chattopadhyay is there? Please,
yes,
please.
Yes.
The question. Sure.
Thank you very much, sir. I would also like to thank Professor Amit Ghosh for his illuminating, if slightly with some pungent wit, his brilliant little talk. Thank you. Now we have a question from Mr. Oyon Mukherjee. He asks, what is the chance that the CRISPR-based editing will be reflected in the phenotype? Practically, how the epigenetic effect, if any, is nullified after editing the genome? Maybe suggest if Dr. Bandyopadhyay would like to take this question.
Okay. So before answering the question, just one comment that I did not work in Jennifer Doudna's lab. In my previous two jobs, I used to collaborate with her. So I know her from there. So the thing is, in one of my slides, I showed that epigenetic regulation can also be modified by using CRISPR-Cas9 system. And and all of the traits or all of the phenotypes are not regulated by epigenetic players. So there are many phenotypes can be seen without any epigenetic interference. And if there is any specific epigenetic interference, then epi-CRISPR is another one field where modifying, I briefly touched that, modifying CRISPR-Cas9, modifying epigenetic signatures by CRISPR-Cas9 can be used.
Thank you, Dr. Bandyopadhyay. Now we have two questions actually from Dr. Aryadip Raichaudhuri. He is from the Department of Biotechnology, St. Xavier's College, Kolkata. His first question is it possible to ensure transplastomics through CRISPR-Cas technology? In other words, is it possible to ensure either nuclear transformation or chloroplast transformation through this technology? If yes, how and what are the reports available till date? Dr. Bandyopadhyay, please.
Okay, so chloroplast transformation by CRISPR, many publications are already there. You can just find out there is a new recent paper in Plant Biotechnology Journal where I forgot the name of the scientist, but yes, there are many. And in this context, I'd like to mention that very recently, David Liu's group from Harvard, they have shown mitochondrial targeting by CRISPR-Cas9 and CRISPR-Cas9 type system, which is a very small size bacterial toxin protein, can be modified to target mitochondria as well. So the answer is yes, there are many examples and of transplastomic by CRISPR-Cas9, and it is available. I'm sorry, the name of the scientist is not in top of my head at this point.
Thank you. The second question is from the same person, Dr. Aryadip Raichaudhuri of Department of Biotechnology, St. Xavier's College. His second question is, how far success has been achieved with regard to environmental stress tolerance in plants, especially with respect to salinity, which is a multigenic trait?
Professor Sopon Datta can give answer na. Sir, this
I think Professor Sopon Datta would be a better person to answer this.
Can you repeat the question?
I understood the question. Actually, Anindo himself worked on it. With the stomatal aperture, he showed a slide that water use efficiency as well as those kind of things related to osmotic adjustment can be made. And even multi-gene trait, they can be also put in one cassette, and that can be also used. So I think it's a complex trait, and it not be as simple as one single trait. But I'm sure it is possible. But Anindo may highlight even with concrete or more example if it is published.
No, sir, you are absolutely, you already gave the answer. Yes, complex traits are really difficult, and we need to know the biology very precisely so that we can modify the genes involved in one particular pathway properly. So it's still a little bit challenging.
In future, if there is a specific transcription factor gene available, the master gene that can, in a cascade way, that it can drive other genes which are linked to this, or as a QTL, linked to this osmotic adjustment for salinity tolerance, it is possible. And Arjo, they've also worked in my lab. Arjo is basically working on salinity tolerance, so that's the reason he probably more interested to know how CRISPR can be used for accelerating that particular work. It's a good question, right?
Okay. The next question is from Professor Rita Ghosh. She thanks Professor Bandyopadhyay for the excellent lecture. Then she says, use of CRISPR-Cas9 editing of genome has been exploited for removing whole chromosome in vitro. this could be of immense potential in genetic disorders related to aneuploidy. How far is the progress in this area clinically? I would, shall we ask Professor Dotto, or shall we go back to?
No, I think if Anindya knows it, I'm no idea about this whole chromosome elimination by CRISPR. I have no idea about that.
Okay, then Dr. Bandyopadhyay, please.
Yes, whole chromosome elimination, few targets were there. And also, if we remember the previous activities of artificial chromosome development, so yes, some activities have been done, but still large chunk deletion by CRISPR-Cas9 is challenging, and whole chromosome deletion definitely is not easy. So some activities are happening in large chunk deletions, but whole chromosome deletion is still challenging. So
one point I just may add that in conventional breeding, Gurudev Ghosh at ERI, he developed some of the lines that eliminating not only one chromosome, several chromosomes, and nanisomic and several sort of things have been developed. But that's it's not desirable, or it's not something but particular trait development. It just happened, and then through selection they looked certain line that can be utilized for eventually for other breeding purposes. So, but with the genome editing, my understanding is it would remain a big challenge to get this kind of work done.
I think such work has been done in the laboratory. They have tried to delete the Y chromosome, and it could be useful for treatment of Down syndrome or the Klinefelter syndrome. So, but I'm unaware this kind of elimination has been done in the lab in vitro.
In vitro, it is possible. In vitro,
yeah.
Definitely in vitro, there's a lot of work has been published.
Whether it is there has been any progress in clinically, whether it is any work, but I'm unaware of that. That's why.
I have personally a question for Dr. Ghosh, Dr. Amit Ghosh. Can you cite any kind of simulation of this process? I mean, has there been any simulation done, either Monte Carlo or molecular dynamics?
I think the best person to answer that question will be Anindya, but I'm not very sure also whether any simulation has been done. But all I know from the literature I follow that basically the bioinformatic analysis to answer a variety of questions is constantly being done. But I do not have any knowledge of the particular question that you have posed to me. Maybe Anindya can answer, because he works in this area.
Yeah, so I mentioned briefly that exactly not simulation, but different CRISPR strategies can be predetermined by bioinformatics analysis or in silico tools, and there are tools available for it. And recently, these new tools are coming out like Sprout or Indelphi, where outcome can also be predicted, or simulation for outcome can be made. So accordingly, we can design the whole experiment, starting from strategy development to outcome prediction. We can bring it in silico platform. So that's possible, but inter-simulation.
Yes, but what you're suggesting or what you're citing is something. It's like a QSAR type sort of modified thing. What I'm looking for is a kind of atomistic understanding of the process, if there is any.
I don't think it has been done, but I'm not very sure because I don't get into it very deeply. But from my reading of the literature, I don't think it has been done as yet. I'm not sure though.
Yeah, I can't answer that either. So I know structural biology, people are working in this area, but I don't follow so keenly that side.
Okay, thank you. There is this one question from the organizing team. Shubhendu Chattopadhyay from YouTube asked, "Sir, can we prevent COVID-19 infection through gene therapy?" I think anybody, Dr. Bandyopadhyay.
There are different experiments going on in this area. So COVID-19, it's a virus. So the RDRP gene and other related gene, capsid gene, etc. People are trying to knock out by CRISPR-Cas9. And also once it enters the body, different RNA targeting systems are also used. We know that Cas13a, but all are in experimental stage, very early experimental stage. So very difficult to predict.
I have a more simple question. Okay. Why, of all caspases, why Cas9?
Cas9, I would say first, it's most studied. A single protein. Other caspases, multiple proteins are there, gene families are there. This is a single gene protein. So, and modification of its different lobes are easier. And many modifications gave predicted outcomes, and it became popular. So, but side by side, there are new ones coming up, like CPF1, which is known as Cas12a. That's also working very well. The very small sized ones are coming up. So it was studied earlier, easy to handle, single gene product. So it was widely used and very popular. But there are others as well, coming up quite fast.
Thank you.
Professor Ghosh, you have to unmute.
I think Amita was probably trying to say something. I'm not sure.
No, what I was saying is, what Ananda said, you know, part of it is historical reason also. They got it and then it worked very well. So therefore, they pursued this path. But right now, the other are also being tried. And some success is also there. But it always happens that you get into the first thing, it works. So you try to push the boundary through that so that you can go. Just like in molecular biology, almost all the work was done with E. coli. Because you wanted to push the frontier, you did that because a lot of things were known. And then, of course, now people are going for other microbes and discovering new things. And discovering not everything is E. coli. Same thing here.
Thank you. There is one question from Dr. S. Roy. Is there any case of off-target? I think whoever it is wanted to say that whether there was any case of missing it, probably.
Let me answer this one. This is a very common question we get in all seminars. So I was expecting this question to come. All these CRISPR proteins are having off-target activities. So apart from your target region, there is a chance that it can also target some other region. Now, it depends on the designing of gRNA. So there are many new algorithms are there which people use so that you can predict accurately where it will go, whether single place or multiple place. So these are available. And many new modifications and synthetic modifications made some of these CRISPR-Cas proteins extremely powerful, but still extremely powerful to target a single domain, single locus. But in cancer cells and certain neoplastic cells where the whole cellular pathway is quite messed up. It's still not very easy to use CRISPR-Cas9 in 100% predictability. But for agricultural purpose, for plant cells, etc., the present modified versions of Cas9 and CPF1 is quite accurate. And there are ways to find out whether your product has any off-target activities or not. So not only making your product or making your strategy foolproof, but also there are ways to find out, like Circle-seq and different other technologies, ways to find out whether the experiment you have done, whether it has gave rise any off-target or not, so that you stop your experiment or you continue or you change your strategy.
And at the same time, this is normally not desirable for a regulatory point of view. So people try to avoid any unintended effect or unintended insertion. So, but it happens, as Anindya says. But one has to be careful that if it happens, one need to know that what it happens, how it happens, why it is not targeted specifically.
Thank you very much, both Professor Dotto and Dr. Bandyopadhyay. There is a question from Mr. Arnab Banerjee from YouTube. He thanks for the excellent lecture, and he asks, is there any update about Pac-Man in genomic editing? Is there any updates about Pac-Man in genomic editing? Would Dr. Bandyopadhyay like to take this?
Probably he's asking about Pac-Man sequencing for genome editing, right? If
I think that is what is asking about.
Yeah, so downstream, once you edit your genome to check the editing whether happened or not, even if there is any off-target or not, different sequencing strategies people took. And nowadays, Pac-Man is quite widely used for those analysis, downstream analysis. So yes, people are using analysis purpose.
Thank you. Thank you. We don't have any. Okay.
I hope we are done.
Dr. Anusua Malik of Envis Center is asking, is there any brand of golden rice? Professor Dotto.
Is the question any brand of golden rice? Golden rice itself a brand. There is nothing golden rice. Because of the carotenoid pathway, color is a yellow orange, and that's the name has just came as a golden rice, and that's it. That itself a good brand name, unless we find another good name. When Bangladesh would be commercially released, the golden rice, probably we would know it, how it commercialized and how people perception change with the availability of golden rice.
Thank you very much, Professor Dotto.
Hope the question answer is finished.
No, there are no more questions, sir.
No, then you end of the part by vote of thanks. Professor Chattopadhyay, we can finish our part.
Yes.
Sure.
Thank you very much, sir. First of all, I would like to thank our Vice Chancellor, Professor Shankar Kumar Ghosh, for being the inspiration behind this webinar. In fact, he prods us, he urges us to do things, to do new things for the betterment of the whole university. We sometimes barely manage. Thank you very much, sir, for being behind us and inspiring us. we would certainly like to thank the luminaries who presented in the webinar today. First, we would like to thank the Dr. Anindo Bandyopadhyay, who is the Vice President of Reliance Industries. He must be a very busy person, but he took time off to give an excellent presentation in in this evening for us. We would definitely like to thank Professor Shapon Dotto, who has taken his time to be with us and has given us a very illuminating presentation and introduction to the whole thing of CRISPR-Cas9. Thank you very much, sir. We definitely would like to thank Dr. Amit Ghosh, who has used all his expertise over the years to summarize the entire system, and yet he presented new insights, some new views to look at the thing from another perspective. Thank you very much, sir. We would definitely like to thank the Dean, Faculty of Science, and Dr. Rita Ghosh, Coordinator of DST PERS, and all the other dignitaries, interested students, faculty members, and all members of the audience who are present. Thank you all very much for making this webinar a success. Thank you.
Just one announcement, and we are going to go for another promotion of science in 2020 Nobel Prize in Physics, the black hole. Hopefully first week or second week of November, we want to go for the black hole for the next time. In this way, we can continue the entire portion on that part with this announcement that they can arrange on that part. With these few words, I thanks again once more to the NACI and the INSA people who are also viewing in YouTube and present over there. Okay, thank you.
Thank you.
Thank you. ভালো থাকবেন
সবাই ভালো থাকবেন
সবাই ভালো থাকবেন ভালো লাগলো খুব সুন্দর কাটলো আজকের দিনটা
রাইট
ভালো লাগলো
খুব ভালো
অনিন্দ থ্যাংক ইউ
Thank you.
অমিত দা থ্যাংক ইউ ওকে